The article Origin and Evolution of Animal Appendages aims to support popular ideas and theories that appendages such as arms, legs, wings, and feet in a variety of animal species have all evolved and adapted for different functions and circumstances over hundreds and thousands of years. Further research into this topic led scientists to a few conclusions. The first one being that the DLL gene, also known as the distal less homeoprotein, was visible not only in the developmental stages, but also later in distal regions of arthropod limbs. The next gene that was to be studied was the DLX protein, which is the distal less homeoprotein present in vertebrates, and this was to be seen evident in a range of appendages including early development of mice, chicken limb buds, and it was also later noticed in the apical ectodermal ridge of the same limb buds. These conclusions left scientists puzzled as the general anatomies and evolutionary histories between vertebrates and arthropods vary so vastly. This leaves two possibilities. One is that the development of the DLL and DLX genes are completely coincidental, or two, that this represents some relationship at a high level as body wall outgrowths. With this information only gathered from two highly divergent taxa, it is hard to determine between the two options. That is why scientists from this article have examined the expression of the DLL and DLX gene in the outgrowths of other animals by using antibodies which specifically recognize both DLL and the DLX proteins. Scientists then went on to conduct their own experiments by using antigens which picked up on the presence of both DLL and DLX genes. They followed previous methods from other experiments, but made a few modifications within the chemical formulas. The results from these experiments led to some interesting conclusions. They found that the DLL gene is expressed in animal appendages amongst a wide array of phyla, not just vertebrates or arthropods. It was found later in developing annelid parapodia, velvet worm lobopodia, and echinoderm tube feet. They decided that although it is possible that these different phyla could be convergent, the chances are extremely low, as it would require individual co-options of the DLL and DLX genes in evolution. It is more possible that the DLL and DLX gene expression is due to a common ancestor and has been used to pattern body wall outgrowths in a variety of organisms. These results, along with a few more statistics, led them to come to the conclusion that animal appendages all eventually trace back to a common ancestor, and that the DLL and DLX genes have come about due to evolution and convergence.